Okay, so I'm Justin Baker, the manager of the Scientific Computing Data Analytics team at CSIRO. Welcome to this afternoon's keynote session. Before I introduce my colleague, my CSIRO colleague, Denis, please remember if there are any technical difficulties, click on the red live support button at the top right. And if you have any questions, post them in the live Q&A box, which is on the right-hand side. And now I'm pleased to introduce our keynote speaker. Dr. Denis Bauer is CSIRO's Principal Research Scientist leading the Transformational Bioinformatics Group. She's an internationally recognised expert in machine learning and cloud-based genomics, having keynoted AWS Summit's Open Data Science Conference, which was in India, and an international conference on bioinformatics in Indonesia. Her achievements include developing open source machine learning cloud services that are used by 10,000 researchers and contributing to Australia's COVID-19 response. The title of Denis's presentation today is very topical: How Computational Science and Data-Driven Health Decisions Help in the Fight Against COVID-19. So over to you, Denis.
Thank you very much, Justin. And hello and kia ora, everyone. Welcome to my home, which is on the traditional land of the people of the Eora Nation. And I would like to acknowledge their elders past, present, and emerging. So with that, let me share my screen and let's get this started. So I would argue that data and technology has become as vital as the healthcare workers themselves. Now, this is a bold statement, but I would like to think that we can back that up with our track record in that field. So I'm working for CSIRO and specifically the Australian eHealth Research Centre, which is the largest digital health initiative in Australia and has the vision of bringing digital technologies and services into the clinical practice. It is actually worldwide quite unique in covering this full value chain from basic science all the way up to services and technology that are delivered into the healthcare service, and then evaluating what the impact was of those technologies. So with that, we are privileged to work with the clinicians, and that technology is something that really enables their work, is something that we hear over and over again. So with that, let's throw to the first question, because we want to make this interactive as possible. And the first question is around, what do you think how data-driven have current public health decisions around COVID been? So with that, you should be able to see the question probably on the right-hand side of your screen, and just go ahead and answer them and see what you think. Okay, you have 21 votes have come in already. Let's go to 30 and start sharing. It's still going strong. It's like this popping when you make popcorn. Pop, 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 pop. And you sort of wait before it actually goes down before you take it out of the microwave. I think this is the time to take it out of the microwave. So share results. So most of you think that the decisions have indeed been based on data. And I think the next bit of information that I share with you backs that up. Back when this whole thing started, I think it was in May, the UK has announced that they're going for a herd immunity strategy. And that was based on their modeling of what is going to happen. Now, there was a paper from the Imperial College of London that estimates that with that strategy, in UK alone, there will be 500,000 deaths, and I think in America it was something like 2 million. So clearly this wasn't a good strategy, and everybody realized that. And the reason for that was that their model was based on influenza, which is a poor representation of how COVID is actually behaving. So I think from that, seeing that with new data that has come in, the models were improved and the results, were something that nobody wanted to accept, the strategy has changed from herd immunity to suppression. So in Australia, We also work with data heavily, and this is what the state and federal government is actually using. So we developed one of the dashboards around the data showing the spread of the different states and the properties of that. And this dashboard was informed by modeling work that has been done before around hospital bed utilization. And of course, we also helped in the mobile health trend, where rather than going to the doctor, it was advised for a long time to actually have telehealth presence with the doctor in order to stop the spread. And the most exciting bit, from my biased opinion, is of course our genomics work, which I'm going to talk about more in this presentation. So with that, let's have a look at an overview of the presentation. There I would like to tell you 3 stories. The first one is around genomic data and how we use it to really inform decision making. Then go one step further and look at general health data and how this will be used really to inform the science that we'll be doing going forward. And then taking a step back from an academic world, our view of the world, to a more economic world, and ask the question, well, how can we build more robust economies? Because clearly a pandemic like that has disrupted quite a bit of the economy. And is there a way of building something that is more resilient? So with that, let's go back and bring us all on the same page around what COVID actually is. So COVID, the disease, is caused by SARS-CoV-2, the virus, which is an RNA virus, coronavirus, and it has 30,000 letters in its RNA, which is basically the blueprint of the virus. It is an obligatory parasite, which means it's actually not a living thing. It needs to hijack your cell's reproductive system in order to reproduce itself. Therefore, it binds to the receptor of the cell surface, injects its genetic material into the cell, and then hijacks the copy mechanism within the cells, the protein mechanisms within the cells in order to build the copy mechanisms of it, and ultimately build little particles, injecting new RNA into it, and then bursting out of the cell, infecting other cells in your body, and then ultimately other individuals. So with that, the question really is, how can we beat it? It's such a new thing. It's something that we can't see. It's something that we have hardly studied in the past. And clearly, the answer to that is developing new interventions. And that will be around diagnostics, around therapeutics in order to treat it properly, and it will be around vaccines in order to effectively stop it. So specifically with the vaccine development, it is quite an involved process. So here I'm showing the overview slide that showcase how CEPI, which is an internationally funded consortium, as well as CSIRO is working together in order to test the vaccine process. So for this to happen, the virus needs to go into the high containment facility that is down in Geelong, which is the only PC4 facility that can have large animals in that containment area. In there, the virus is replicated, it's sequenced to really better understand it, and animal models need to be developed in order to test it. From there, the preclinical testing is happening to looking at the safety and efficacy of the work. From there, the clinical trials are happening, which we've heard now with the AstraZeneca Oxford vaccine that is currently in stage 2, and there are some hitches at the moment, but we'll see how this goes further. But ultimately, once those clinical trials 1, 2, and 3 are successful, it goes out into the general public. It's similar with drugs and diagnostics as well, in that it needs to be pre-clinically tested in the lab. So, as if this wasn't complicated enough, there's one other thing that makes it really difficult, and that is the COVID is mutating. Like any other RNA virus, it is mutating as sort of part of its natural evolution. And I'm showing here the different strains or the different versions of the virus that are currently known. So these are single, so out of the 30,000 bases of the RNA virus, each one of those can mutate And each one of those could have a functional consequences. So people are recording them and categorizing them in order to compare one virus to the next. So as you can see, there are currently 1, 2, 3, 4, 5, 6, 7 strains that are versions of the virus that were recognized and tracked. And some of those have the D16.4G mutation in it that you might have heard of, that is currently investigated whether it's spreading the virus more easily. So from there, another question to you: how fast do you think coronavirus is mutating relative to something that we already know, relative to the influenza virus? So is it mutating faster or slower? And let's start that question now. Oh, it looks, currently it's 50-50. Oh, this is interesting. Okay, let's share the results. So you're saying it's not, okay, now it's faster than influenza, which is interesting because it is actually mutating slower than influenza, which is a good news story. So influenza has something that is called recombination in it, where basically it can shuffle its genetic material quite substantially, which means from one season to the next, its envelope, like how it's packaged up, can look very different and our cells do not recognize it, which means that every year we need a new vaccine. Whereas the coronavirus does not have quite such an aggressive reshuffling mechanism. It is actually quite faithful to its template. It has a proofreading mechanism, which is one of the few RNA virus that does have a proofreading mechanism. So therefore, my analogy is like influenza is when you bake cookies, you have the option of how to put it into shape. You can roughly cut it with a knife, and that's basically what influenza does it. So if there's a star roughly coming out of it, 8-armed, 9-armed, as long as it sort of looks like a star, influenza is happy with it. Whereas coronavirus is very particular about it. It uses a cookie cutter in order to replicate itself properly. And therefore, the changes that are in the coronavirus, the mutations that are in there, are at a much slower rate. But it is mutating. It does have the capability of mutating, and especially as something that has only recently jumped into the humans, it is under probably a larger evolutionary pressure than something that has already been there for quite a while. So therefore, it is thought to have originated from bats. There might have been an intermediate host, but eventually it came to humans. And then from human to human, it is spreading, accumulating potentially new mutations. So those mutations could cause it to be more infectious, for example, or it could cause more severe disease. Therefore, the question is, which one of those versions of the virus do we want to build the vaccine for? And therefore, the two questions that we wanted to answer is, does these mutations hinder vaccine development, and does it cause a more severe disease? So with this, we developed a new computational methodology in order to fingerprint this virus. So with this fingerprint, we can uniquely identify it. And therefore, when we compare one virus to the next, it's like a lineup of suspects. We definitely know exactly which one we're looking at. So therefore, we can determine the similarity between two viruses, and then from there use machine learning in order to put it on a 2D map where the distance between two viruses represent the evolutionary distance between them. And since there's a technical audience, I can go into a little bit more detail. So what we did is we take the sequence, we create little k-mers, so that's letters or words of length k, and we record all the unique k-mers in a sequence, which then becomes our number profile, our profile that fingerprints that virus. And from there, we use principal component analysis to reduce this high dimensional space down into a 2D map that we can visually look at. So we've applied this to all the coronaviruses that were available back in May, which were only 200. But mainly what we show in there, which is basically the yellow dots, is basically the full spread of the coronaviruses that were available at that stage. But then we color code the ones that were used in order to test the vaccines against. And the question we wanted to answer is, where do they fall on this 2D map? Do they represent the ones, the major spread of the virus, the major evolutionary space that it has already accumulated, or are they weird outliers that no one should care about? And the good news again is that the versions of the virus that were chosen by the vaccine testing facilities are actually quite representative of the spread at that time. When I say at that time, clearly, 200 isolates that were submitted back then, this has dramatically grown. Therefore, we do need to have a continuous way of analyzing and reanalyzing this data to really evaluate whether the current ones that the vaccine is tested against is actually something that stays relevant and representative. So therefore, we've built an online, a cloud-based platform to reanalyze that data. And of course, we're using a semi-serverless architecture that only consumes resources when it's actually running, and when it's not running, when it's idle, it doesn't cost anything. So it's a very resource-efficient, scalable way of analyzing the data. So currently, 1 out of roughly 200 confirmed cases is sequenced and is submitted to that resource, which is quite an interesting progression, and it's probably the largest data set of any sort of infectious disease in the world or in history. So this will be an interesting thing to analyze further. And what that actually means in real numbers, currently we have roughly 90,000 isolates from around the world where the sequence was determined and was submitted. So therefore, we have 100,000 roughly times 30,000, which is the length of the virus, as data points to analyze, which is quite substantial. So with this, given that it's so substantial, it has thrown up some problems around the computational feasibility of that analysis. So one was that the aligner, which is the way of putting the physical sequencing material onto a reference framework, was breaking down because of memory issues. Because if you analyze, or if you're trying to run over and over these 100,000 sequences, the aligner is just struggling with that. Similarly, with the principal component analysis, in that we had so many spurious changes in those sequences that the principal components were not really picking up the main change anymore. Like the major, or the largest differentiation wasn't explaining that much of the variation that we're seeing in the data, which means that principal component analysis for doing the dimensionality reduction is probably not going to be a sustainable way going forward. Similarly, with our visualization page, that just having 100,000 data points on our interactive map or interactive picture, it was just taking too long to load. So we do have to do some optimizations around that as well. So currently it is working, but in order for this to be sustainable in the future, and for other outbreaks where probably the sequencing will come in much faster, we do need to develop new methodologies. So with that, coming back to an audience question, do you think the mutations do actually have clinical impact? Now, people are saying that the coronavirus, and these are headlines from news outlets, coronavirus mutations have made the virus more contagious, and is the coronavirus really getting weaker? So what do you think? Do those mutations make a fundamental change in the clinical behavior or not? Okay, sharing results. So you're leaning towards yes. Now, the World Health Organization, being as conservative as it is, was saying that there is currently no evidence that it is mutating meaningfully to become more lethal or more contagious. Now, this was in June. Therefore, this is very much an open research question that we want to address as well. We do know that there are location-specific mutations. So, for example, looking at the Australian dataset, we were able to see a mutation that is specific to Victoria, and that was back in April when we did that analysis. So this mutation is in 5%, was seen in 5% of Victorian samples, and were not seen anywhere else in Australia, and only 0.3% in the world. So with this information, of course, you can then backtrack the origin, the likely origin of a mutation. So for example, when you have a virus-free, like New Zealand, a virus-free land, and suddenly a virus comes back in, you can then compare its sequence to the world sequence and potentially pinpoint where that particular virus might have been picked up. So this brings us back to, and then tracking specific mutations. So here we want to track one mutation out of the 30,000 letters in the genome and do that as efficiently as possible. And not only that, we also want the data to be secure because the data was created with enormous effort, so we can't risk that the data is reversely identified. But at the same time, we want to bring in as many data sets together as possible. And again, we created a serverless, cloud-based architecture for doing exactly that. So this serverless COVID beacon allows data to be shared, from individual mutations to be shared across multiple data sets. And here the example is, of course, the D14G mutation, which is currently investigated whether it is able to spread the disease more easily. And what we see here specifically for that mutation is that it has become the most prevalent strain or version of the virus that is out there. Like it started back in, like what is shown here is that in December, there was none of the sequences showed that mutation. And then January, February, starting to pick up specifically in Europe. And then now it is the dominant version around the world. So if anything, certainly we can't conclude from there that it makes the virus more contagious, but it certainly might be a founder effect that it was part of a super spreading strain or chain, super spreading chain, and therefore had more opportunity to spread. Either way, it is now for all intents and purposes, it is the pandemic is dominated by this particular mutation. And our technology and our tool allows us to track the frequency of this mutation as well as any other that might pop up in the future to be clinically relevant. So, but coming back to the question, can we find something that is actually making the disease worse or better and has functional consequences? So with that, we're using one of our technologies that we developed in the human health space called Variance Bug. So Variance Bug was originally designed for detecting disease genes in human cohorts, so human case control studies, which means creating a cohort of people with the disease and creating a control cohort of people without the disease. And the question was, can we find the molecular origin that drives the disease? So particularly we partnered here with the project MINE, which had 22,000 genomes from patients suffering from the motor neuron disease ALS, which is the disease that Stephen Hawking suffered from. And here the question was, can we find, or the basic premise of an association test is basically that you have the genomes of the different individuals. You then identify the mutation, which is here represented by boxes, that make that person unique. So on average, there are 2 million differences between you and the next person. Then you group them into people that have severe version of the disease for COVID or mild versions of the disease of COVID, and then identify which ones are prevalent. So for a virus, that would mean listing up all the virus sequences, identifying the changes that they have, and then seeing whether there's one location in its genome that is enriched in the ones that have the severe disease compared to the mild disease. Now, in human genomics as well as in viral genomics, it's not as simple as that. It's unlikely that there's one location that drives a change. It probably will be locations working together. So there might be one mutation that causes, an effect, but there might be other locations in the genome that cause a counter effect. So it's basically this balancing, or it might be versions where it's not a counter effect, but it's an amplification effect. So it's making the originally changed even more dominant. Therefore, when we analyze this data, we need to analyze it as a whole rather than each individual location separately. And this is basically exactly what Variance Bug is doing. So Variance Bug is a random forest machine learning approach that builds decision trees, thousands, tens of thousands of decision trees based on the data that we have. Now here, what we collected from the international data set was 300 samples of people with severe disease and 200 samples of people with mild versions of the disease, and ran Variance Bug over it in order to find whether there are any functional consequences to it. And what I'm showing down here is stretch the whole area of the virus, and then as spikes, I'm showing the locations in the genome that were associated with the outcome, so mild versus strong disease. And it's interesting to see that here the D614G mutation again is coming up as potentially being associated with the outcome of the disease. And this is interesting because this particular mutation is in the spike protein, which is the one that actually binds to the receptor of the cell for it to inject its genetic material to it. So it's entirely plausible that it could have an outcome on the severity. However, this study is hopelessly underpowered. Like 500 samples is not going to give us the statistical power that we need in order to differentiate 30,000 positions in the genome. All of this could be just random chance, but it is a trend and it's a methodology proof of principle that if we have, or once we have the right amount of data, we can do this analysis, and this will help determine whether viral changes can actually have an influence on the disease outcome. Another element to that is that the healthcare data is just not there. Well, the reason for that, we do have 80,000 samples already, as I said in the past, but only a fraction of those actually have the severity of the disease annotated. And here I'm showing the word cloud of what is actually annotated to it. Mostly it's unknown, not provided, hospitalized, released, which is a meaningless tag to that sample because it could mean anything. Similarly, with how it is annotated, it was, when we did that analysis, it was a free text field, which means clinicians could enter anything they wanted. And specifically for example, the loss of smell could be annotated in a variety of different ways. So what I'm showing here is from SNOMED CT, the hierarchy or the ontology linkage between the different terms. So any one of anosmia, congenital anosmia, yada yada, is linked to sense of loss of smell, which is a subset of finding of sense of smell. And then it goes all the way up the tree. Therefore, depending on what the preferences are of that clinician, they could choose any one of those locations along the tree to annotate that that sample, which any one of which is academically or medically accurate, but the machine needs to have that single term that links it all together. Therefore, there needs to be a different approach for that. We do need to use FHIR terminology, which is the fast health interoperable standard that is emerging in order to allow people to still enter free text, but then automatically be linked to this particular ontology. And therefore, and this brings me to the last bit of the presentation around robust economies. So let's throw back a question to the audience. And this one is a bit different. It asks, post-COVID, what do you think is the most important thing? $33. Oh, wow, 40. People are getting almost have 50 votes. Well, this is probably the most answered question out of all. Well done. Share results. So resilience. Now I appreciate that probably I was a bit leading with that. I think all of those are important going forward. As in, we do still need efficiency in order to bring our economy back. They need to be more resilient because there will be pandemics in the future. We do need to prevent really from pandemics in the future actually happening, and we do need to work better together. But for this particular talk, where I was driving at was that Gartner, which is a consulting company, was saying that post-COVID, designing for resilience is more important than designing for efficiency. Because obviously, if you can't operate because you're disrupted, then you might be as efficient as you want. If you can't do anything, that's not helping. So our answer to that, how to get more resilience, is to go to the cloud. So what is the public cloud? We think it's so it allows people to really access the latest compute technology without upfront investment. Typically, when you go to the latest newest technologies, like GPU, for example, you have this massive cost of creating or installing a GPU cluster before you even can work on it. With the cloud, you can just rent that technology, see if it's any good, if it's useful for your use case, without breaking the bank or without having a liability on your hands. Because if it doesn't, you can just stop using it. It is much more cost effective because you can scale easily. You can start with small use cases and then scale up once those use cases have been demonstrated or validated. And of course, it allows you to be more connected to the world, because if you spin up a solution in your region, you can easily replicate that version or that infrastructure that you created in your region and put it into a different region. Like, I'm sure as computational researchers, we've had more than once the issue of lag time, that if you wanted to access a resource on the other side of the world in Europe or the US, it would be much more complicated to do that rather than having something here, which, where the idea of mirroring came from. With cloud, you can do it the other way around. You can mirror, if you want, your technology over there rather than bringing the data over here to process it. So, but coming back to what cloud computing actually means for us researchers. So the way that I think about it is it's like an in-room sound system where you can bring along your device, your tablet or your phone, and you stick it in, and whatever is on your device, you can play the music in the in-room system. And it's a similar thing with the cloud. You can bring your software along. So this is Variance Bug software, and we can just stick it onto one of those virtual machines in the cloud and run it there and use their computational power in order to drive the analysis that we want to do. Similarly, with an in-room system, you can tailor it. So depending on what style of music you like, you might want to have a bit of oomph with it. So you need a subwoofer around it. And similarly with the cloud, you can scale it up or can have even specialized, what I'm showing here, GPU specialized hardware on there to utilize. So another question, so this is an actual question that I have to you. Have you actually used the cloud for research before? Yeah, overwhelmingly, overwhelmingly positive. So most of you say yes or not yet, which indicates that there is a desire for the cloud or the recognition that cloud might be an interesting way forward for research. And I wholeheartedly agree with that. So not only does it allow more flexibility, I think it is more frugal with our research money in that we can achieve more with the cloud. And what I'm showing here is really an example that for us that drove that home. Like with cloud computing, we can save 300-fold of the cost. And with a 300-fold cost saving, you can probably hire a postdoc from it. So specifically here, this was the use case around sharing data online. So it's a beacon, the beacon use case where there's genomic data and you want to make that available for other people to query that particular data set without having to share the data in its clear open form. And there's a consortium in the UK called Genomics England that have 100,000 sequences that they wanted to share. And for them to do that, it would cost $4,000 per month in order to make that particular data set available to the world. Whereas using serverless technology or the cloud technology that we developed, it will be $15 a month. So it's less than a cup of coffee per day to really share that enormous data resource with the world in a way that is fully interoperable without having to give up on the data privacy. So I think this, to us, it was really a no-brainer to do everything we do in that particular way. So coming back to the resilience, quest. We want to offer our technologies to the world. And we want to do it in a way that we don't have to be the consultants that they have to call in order to install that software on their cloud devices. And for that, excuse me, for that, we're using digital marketplaces. And specifically, digital marketplaces allow cloud-based technology, I do have a bit of a cough, but I got tested, so it's all good. Digital marketplaces. So digital marketplaces you basically can think of as apps of in your phone or your tablet, where you can tap an app or subscribe to an app and it automatically deploys in your on your device and you can use it from there. It's the same thing with the cloud, that you can subscribe to individual solutions, in this case Variance Bug, and it automatically deploys in your cloud account, meaning that you have full control over the technology that is actually there because it's in your cloud account. You can tweak and fine-tune it, and it is right next to your data as well, so you don't have to give up on your data. You can keep your data encrypted in its way that it is protected in your account, and you just link the Variance Bug version of it that is under your full control to it, and you don't have to complicatedly install anything or configure anything. Everything is there and is working properly. And it's not only Variance Bug, of course, you can choose from a wide range of different technologies that you can subscribe, and it's automatically deployed in your cloud account. Now, when I say it's not only Variance Bug, it's actually not quite true, because Variance Bug has become the first health technology from a public sector organization worldwide to be on the marketplace. So with that, it's probably the first forte from academic researchers into that space, because typically the marketplace is used to sell products, whereas Variance Bug is freely available and is therefore more driving towards a reproducible research angle, sharing knowledge, sharing the way that data needs to be set up, rather than actually making money from it. So with this brings me back to the summary of the talk, the 3 things to remember, the take-home messages. There's a lot of IT that has been developed for COVID-19, and I think it really has helped. So when I think what will get us out of this pandemic faster, my answer to that is definitely technology. And healthcare, through it, healthcare has become much more data-driven. There's this joke that says, well, who was the change manager to drive the digital transformation? Was it the CEO? Was it the CIO? Or was it COVID? So with this, I think there was this urgency that things needed to change. The business as usual was not acceptable anymore. And suddenly, things that were stuck in their way have become shaken loose, which I think is a good thing. But there is still a way to go. Like we will not have an electronic medical system, which is really what is necessary in order for health to become fully digital. We will not create that overnight. So there is definitely still a road ahead. Potentially helping along that road is the marketplace, which I think of as the way to make us more resilient. It is a way of reproducible research. It's the way I think as academics we could share our setups and our data more effectively. And definitely it should be something that you could consider for as a deployment for your own code. The only thing left to do is thank the team and encourage you to go to our webpage if you want to find out more. And I think we have 15 minutes timed for question and answers.
That was great, Dennis. Thanks for a really interesting presentation, particularly for me, who's part of the lockdown in Victoria due to COVID. So we do have a couple of questions, which I'll put to you. Firstly, from Melissa Evans, and she's asked what kind of aligner you were using. I'm assuming genome alignment.
Yeah, look, they're different. What we're doing here is multiple sequence alignment. And they are specifically ones that are built for COVID. That they're coming from England. But none of them really address the problem properly. So there's, if you're in that space, I implore you to come up with something that truly can, multiple sequence align hundreds of thousands of sequence, which are not tiny little bacteria, but actually, largish viruses that we have.
Mm-hmm. Thank you. And from Joel Pauling, I guess a question that's on a lot of people's minds about cloud computing. Joel said, how do you deal with the seductive lock-in that native services like Azure and AWS, etc., have that are effectively the new vendor lock-in?
Yeah, vendor lock-in is something that is thrown around quite a bit. For us, it's like we were very conscious from the beginning in that, because we never knew whether whether AWS, Azure, Google, any one of those could have taken over back when we started it. Now I think it's very clear that all of them will stay around, and that all of them actually have the same technology. When it comes down to it, they have the same technology, so they are basically interchangeable. which means their solutions have become interchangeable. And the technology that we develop, it is, we're trying to make it as cloud agnostic or vendor agnostic as possible, in that we develop with CloudFormation or Terraform specifically. So Terraform has a claim to fame that whatever technology you develop, you're just building a template for it, and then they can translate it into the specific vendor languages. Because an instance like a virtual machine on Azure should be exactly the same as a virtual machine on AWS or on Google. It just has a different name. The reality to that, of course, is that it is not just the language thing. Like, there are different ways around the way that, say, identity is handled, or security is handled, or communications is handled. So I think the reality is that at this stage, it's not quite as seamless as it could be of developing a technology and then rolling it out across different cloud platforms. But I think there is the general understanding that this is something that they want to arrive at. So there should be a language to rule them all. And you just write in that language, and you hit the deploy button on Azure, you hit the deploy button on AWS, and it just seamlessly gets spin up on there. So I think multi-cloud presence is something that we're going to see much more happening in the future.
Thanks, Denis. So related to that, Joel's also asked, how is Variance Spark delivered? Is it a Kubernetes operator, like operatorhub.io?
It's not yet. So we did have some elements around Docker and Kubernetes in order to deliver it, but ultimately what it is, it's a Elastic MapReduce cluster that we're using, which means that this is already a modular component that in this case AWS has created, and all we need to do is create a bootstrap script that build the head node as well as the compute nodes. And therefore, we don't have to go down the more complicated, even though more elegant, route of Kubernetes just yet. In fact, when we tried it a year ago, the problem was that it wasn't quite as scalable as we needed it to be with the Elastic MapReduce setup specifically. So it would create clusters that had problem around the communications between it. So back then, a year ago, we gave up on that avenue. But if you're an expert in that space, and if you're interested in helping us revisit that particular topic, we'll be very, very, very keen of actually doing that.
Well, there's an offer to keep in mind, Joel, I think. That sounds good. A question from David Hood, which I guess is philosophical as much as anything. David said, because there's a negative relationship between the 2019 Global Health Security Index, which has a tech focus, and the actual pandemic outcomes to date, is doing simple things quickly more important than data?
Yeah, that was basically going to my question, right? Should we wait for the data to be there, or should we just, do something that has an immediate outcome, like wearing a mask, rather than, say, building a complicated tracking system that would digitally help us wear a mask? And I think the answer to that will be in hindsight, and that once all of this will be over, probably it will be evaluated. It's like what we have now with the Spanish flu, where information come out of communities that had a hard lockdown would do much better financially, as well as economically as well as saving lives, compared to others who sort of were driving the middle way. Like that was, but that can only be evaluated in retrospect and in hindsight. So yeah, it is a philosophical question, and I think the answer that we have to that is we don't know and we might never know. I think the interesting bit that struck home with me was the statement of saying, well, if we have done too much, we will never know. But if we've done too little, we definitely will find out. And I think that's sort of something to keep in mind with this.
Sure, thank you. And this next question from Eric. I won't have a go at your surname, Eric. That's probably a bit too much of a challenge for me. But of direct relevance to the RSE community, how can we help the scientific community and IT operators to better understand and learn how to use cloud technologies so that they can take advantage of the benefits that you've described?
Tricky question. That is really a tricky question, because if I had the answer, we probably would be much more successful than we are now. I think it is such a steep learning curve for us as well. Like when we endeavored on this quest 6 years ago, it was baby steps for us. Like we were finding out so many things that even we as computational scientists would not know, would lack the skill set in. And then I was lucky that CSIRO funded this whole learning curve, or the upskilling for us through the research that we're doing. But if you're under pressure to actually deliver a hard outcome, you don't necessarily have the luxury of upskilling in something that is not quite accepted as essential. Because I think this is where we're at the moment, in that cloud is there as something that will happen and that there's a transition to, but is not quite a business imperative or research imperative just now. It will, in my mind, it will become that, because it just makes sense. It will make everything more effective, and if you're not on the cloud, you're just left behind, in my mind. But I think a lot of people don't see it that way necessarily at this stage. And again, hindsight will prove either them or me, right? And so it's really difficult. And I think the cloud providers themselves do not make it as easy as they probably could. I know that both Azure, so all three actually, Azure, Google, and AWS have created roadshows, which are specifically targeted for academics to showcase what people can do with their technology. But I think, it's sort of pitched from a commercial angle. It's sort of coming from it as, well, your business needs to be more scalable, more improved, which rings really hollowly for researchers, because our main objective is to find insights into the data or into a fundamental concept that we're researching, and whether that takes a month, 2 or 3, and ultimately it doesn't really matter, because we have heaps of things to do in the meantime. We can write papers, we can go to conferences, etc., etc. So therefore, this immediacy is not the argument that will gel with us. So therefore, I'm coming in more from the interoperability, from the community angle, that we can build something that is easily rolled out to other researchers and shared with other researchers. And I think, from my perspective, that is something that I would like to see more in the community, that a paper comes out, and rather than having the reproducibility as a GitHub repository, which is, it's a good start, or one of those small code ocean examples, which is good as well, don't get me wrong, but there's still a lot of work for me involved in order to port that to my data. Whereas I want to have something that I can just snap my fingers, it's already rolled out, I point to my data, I press enter, and there I have, I potentially even can reproduce their results, as well as have the reproducibility on my data. So replicate their results or reproduce on my data. So I think, to me, this is a much stronger argument for the scientific community to listen up, rather than the business and the business angle. So I think the short answer to that question was, maybe target it slightly different, have a different argument for why researchers should go to the cloud. That comes really from their angle.
Mm-hmm. Okay, thank you. And I guess a much more specific question from Damien Mather. Have you considered using corpus linguistics-based lexicon supervised text mining to recover quantitative predictive inputs from your text data?
That sounds awesome. Let's talk. No, we do. I mean, the rest of some groups in the eHealth Research Center are using natural language processing in order to process electronic medical records, or semantic question structuring. I don't know what the technical term is, but a way of structuring search terms in a more systematic way in order to really go into the academic literature to find the answer to a question. So those angles, I think, are really good, but the data is currently is not up to a level to actually throw these heavy-duty technologies at it, I think. Happy to be proven wrong. In fact, I'm very keen to be proven wrong, because I think this is definitely where we need to go, and I'm happy to leapfrog to the technical solution rather than the currently where we're at, I think, which is more a people solution or problem of getting people into the mindset that data needs to be digitally collected.
Okay, thank you. And we've got about 2.5 minutes to go, so I'll give you, this one's more focused on the medical side of things. With the initial viral dose, this is from Joe Chen, would the initial viral dose that a person was exposed to need to be taken into account in order for the genomic biomarker analysis to be meaningful? I could imagine a person who is genetically robust to SARS-CoV-2 still getting very sick if they were exposed to a large amount of the virus.
That is an interesting question, and I think I've seen in the literature the question whether the dose of the virus is actually having a larger influence on the disease outcome rather than the mutation of the virus itself. I would argue that the healthcare system, the state of the healthcare system, has probably an even larger outcome than both of those combined. So therefore, this question is really so complicated that, and so multifactorial, that there are so many covariates that are playing into that, that it will be a question that we will be researching for a long while before we actually have a definitive answer to whether the genomic markup of the human, the genomic markup of the virus, or how that person is actually treated, which one of those has the biggest influence, will be definitely a question for the next years to come.
Okay, sure. So a non-trivial question for our remaining 3 minutes, I can see
Yes.
that. We've got about 1 minute to go, Denis. So thank you for a fantastic presentation. I found it really interesting. It's really appreciated that you came along and explained all of that to us. I have a very quick question for you in the remaining 50 seconds. Can we predict which way the virus is mutating? I know we have various mutations or mutants of it. Is there anything we can say about the way it's going? And we may get cut off, but I'll be interested to hear your thoughts.
This is a super interesting question and something that we're looking at as well. So this was the original research that we started with influenza to identify where influenza is actually heading and whether we can predict the next strain that is coming out next year. So we do have some ideas around that, and I think those ideas are applicable to COVID as well. So definitely watch the space, and if you have ideas, get in touch with us, because it is an active research area that we're on.
Okay, fantastic. Thank you very much.
Thank you so much for listening, and talk to you soon. Bye.
Okay, bye. Thank you.